Okay, here we are going into the second section in your chapter here, and we're talking about the cell cycle, which is in two parts. The first part is known as interphase. And really, this is the vast majority of the time. This is over 90% of the time. Normal cell life, if you will, when it's not undergoing division, when it's not undergoing mitosis. So interphase, we're going to find out, occurs in three stages. The first stage is known as the G1 stage. And the G1 stage is when the cells double in size, and cell grows. So we have just normal cell growth going on, kind of nothing special. And then it'll pass a point, and the DNA itself will replicate. Now, what'll happen, we're going to find out in just a second, is that the DNA, that singular chromosome, here, is going to initially look in the G1 stage. If it were to coil up, it won't, but if it were to coil up, it would look something like this. However, after the S stage right here, this DNA, and we're going to learn a lot more about this in the very near future, is going to replicate. So this strand over here is going to replicate into what's called the sister chromatid. These are chromatids. It's a sister chromatid over here bound by what's known as a centromere. We did see those images briefly in the last slideshow, and you see them in the book when it was talking about the karyotype. So we have a centromere in the middle and sister chromatids, but the whole point here is whether it looks like this after replication, after replication we enter what's called the G2 stage, whether it looks like this in the G1 stage, or it looks like this in what's known as the G2 stage. Either way, this is still one chromosome. Twice the DNA over here, but one chromosome. That can be a little confusing. I know when I was an undergrad that confused me. But this is one chromosome here, one chromosome here. Is that this one chromosome has duplicated its DNA, but it's still one chromosome bound by this central centromere. Okay? So once again, going over interphase, this is once again, more than 90% of the time. This is normal cell life. We've just got normal cell doing what it needs to do. It'll keep growing, and eventually, when it knows it's going to replicate and go to the next stage, into mitosis, next phase, it'll be doubling in its organelle, growing in size. It'll replicate its DNA during this S stage, and its DNA will go from looking more or less like this here to the typical X, what we look like over here. All right? And once again, 90% of the time. The other part is actual cell division. And just as interphase had 3 stages, okay, cell division has both mitosis and meiosis. And in fact, mitosis, the main focus of all this, that we're going to be talking about, actual nuclear division, separating that DNA to make sure that the daughter cells have the same in the humans, for example, 46 pairs of chromosomes, right? Or 46 chromosomes, 23 pairs. That's actual nuclear division. It itself has multiple stages, and that's what we're looking at. That's what we're going to be looking at in this chapter. In fact, I'm going to give you a highlight. I like just to memorize PMAT. We're going to find out my little PMAT for mitosis is prophase, metaphase, anaphase, telophase. We'll get there, but just remember for right now, PMAT. That'll help us a little later on when it comes to mitosis. So the big point here, once again, is most of the time, if we were to do this cell cycle, and they always designate cell cycle in this, like this, in a cycle, where most of the time we have interphase broken into G1, then a little time for DNA replication, S stage for synthesis, synthesizing DNA, okay, and then G2, and then we actually get to cell division down here, which will be mitosis. And in fact, I'm going to go put my PMAT now. I'm going around in the cycle, so the first stage of mitosis is a P, and then it'll be in prophase, metaphase, anaphase, telophase, and then it's actually dividing up the cytoplasm, which is kind of its own little part right here, cytokinesis. So hey, that's a much better picture, isn't it? Now remember that this actually is not, as far as this image right here straight out of the book, it makes it look like this mitosis and cytokinesis is, a quarter of the time. This is not representative by time right now. And what we're going to find out is all of these stages are very much controlled by the cell. And there are regulatory steps all throughout. And we're going to find out that, especially like when we come to DNA replication, that there are regulatory steps in here to make sure that we get the copying of the DNA right. Because remember, we have to have an exact copy of DNA from parent cell to daughter cell. So there's multiple steps of regulation and determining, make sure that everything gets right, and in fact that we are going to go all the way through cell division, because some cells, quite frankly, don't divide in the long run. They've reached a certain point and they're going to grow and do their thing and eventually die, but they're not going to divide. They're not going to go all the way through mitosis. So there's lots and lots of regulation here, and actually for the level of this class we're not going to get into it. What I want you to get through in this module are the basics of this cell cycle, that 90% of the time, more than 90% of the time, it's the cell's doing its thing, it's growing, it's doing whatever it needs to do as far as its functions within the human body, whatever tissues and organs they're in. And they'll have normal growth, and at one point it'll make the decision to go ahead and replicate its DNA and enter that S phase, and then it'll enter the G2 phase and eventually that mitosis. Now, if you can just get for right now, once again, the big ticket item here is, okay, interphase most of the time, and then this cell division part is separating out our DNA. So once again, that's what we need to be even identically in the daughter cells, and then separating everything else, which is cytokinesis. Now, your book has a whole bunch of animations. You may have already watched them, but you know what? I'm a visual person, so we're just going to go over some of these animations that it shows us regarding this mitosis and cytokinesis, this whole cell cycle. The first one really emphasizing this whole cell cycle, once again, how it works with interphase and cell division.
Most eukaryotic cells follow a process of growth and division called the cell cycle. These events include a growth stage, mitosis or nuclear division, and cytokinesis or division of the cytoplasm. Throughout interphase, the cell is engaged in growth and metabolic activities. Interphase can be further broken down into 3 discrete phases called G1, S, and G2. During the G1, or first growth phase, normal cell functions occur as well as cell growth. In the S phase, DNA replicates, producing 2 copies of each chromosome. During the G2 phase, the cell continues to prepare for mitosis and cell division. Mitosis, or the M phase, has 4 stages: prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes become visible and condense, becoming shorter and thicker. Each identical copy of a single chromosome is called a sister chromatid. The nuclear envelope breaks down and spindle fibers form as microtubules grow out of the centrioles that move to opposite poles of the cell. During metaphase, the double-stranded chromosomes line up along the equator of the cell. The spindle is now fully formed and the microtubules attached to each sister chromatid. Anaphase begins when the sister chromatids of each chromosome begin to separate. The centromere that holds sister chromatids together divides, and the chromosomes move away from each other along its spindle fiber. In telophase, the two groups of chromosomes reach the opposite ends of the cell. As a new nuclear envelope starts to form around each group, the chromosomes uncoil and the spindle disappears. The division of the cytoplasm and organelles is called cytokinesis, or the C phase. The result of mitosis and cytokinesis is the formation of two genetically identical cells. To summarize the entire cycle, G1, S, and G2 phases occur during interphase while the cell is growing and preparing to divide. Mitosis, or the division of the nucleus, consists of 4 phases: prophase, metaphase, anaphase, and telophase, and in these phases duplicate copies of the chromosomes are split equally. Finally, the cell's cytoplasm divides during cytokinesis and results in the formation of 2 genetically identical daughter cells.
Okay, so that was a bit more detailed than I went over because it actually did go over the basics of mitosis, which you will need to know and understand by the end of this chapter, but I wanted you just to see it, see how this whole cell cycle works. So once again, here's another animation that's referenced in your book, and honestly it's more of the same, just a little bit of a different viewpoint. But once again, I just wanted you to see it, so let's go ahead and hit play here.
Mitosis is a process of nuclear division by which replicated copies of a cell's DNA are organized into chromosomes. The identical copies of the DNA are then divided equally between 2 daughter cells. Early in mitosis, during prophase, the nuclear membrane and nucleolus begin to break down. As prophase continues, the DNA condenses to form chromosomes while the mitotic spindle, made of proteins called microtubules, begins to form and attaches to the kinetochores on each chromosome. During metaphase, the chromosomes are moved to the central plate of the cell by the spindle microtubules. In anaphase, the microtubules attached to the kinetochores of each chromosome shorten, drawing the chromatids of each chromosome to opposite ends of the cell, while the unattached microtubules elongate, stretching the cell. During telophase, the chromatids, now called chromosomes, reach the poles of the cell as the unattached microtubules continue to elongate. The nuclear membranes begin to reform as the nucleoli reappear and the chromosomes relax as the DNA unwinds. Telophase is followed by cytokinesis, or division of the cytoplasm. In animal cells, a cleavage furrow forms at the center on either side of the cell, pinching through until the original cell is divided into 2 daughter cells. In plant cells, cytokinesis is different due to the presence of a rigid cell wall.
So once again, more detail than I've lectured already about, but just giving you a preview of what's to come as far as in the detail of mitosis. And yes, if you've already read the chapter, you know this already, but yes, you do need to know each of these individual steps of prophase, metaphase, anaphase, telophase, and everything that goes on in there. So, moving on once again. Now, this little animation is going to talk about control. Now, remember, I said throughout the interphase and that there has to be regulation, making sure that the cell is going to go all the way through mitosis and actually replicate and have 2 daughter cells as a result of going from a parent cell to 2 daughter cells. Because once again, there will be points. There will be times when cells will not do this, and pretty much their cells are going to finally just do their job and die. So there are regulatory points to make sure all the way through that the cells are, one, going to go all the way through this cell replication process, but also two, that the replication process is going right. Because if there's a problem in the DNA or what have you, when it's replicated in the S phase, well, then there will be a problem with the two daughter cells. So there's always all this regulation throughout, and that's what this animation talks about.
The process of cell growth and division in eukaryotes is called the cell cycle. This cycle is divided into phases based on what is happening in the cell at a given time. A cell grows during the G1 phase. During this phase, there is a chemical checkpoint that controls whether the cell will divide, delay division, or enter a resting stage. When conditions in the cell are right, the G1 checkpoint will be passed and the cell will enter the synthesis S phase. During the S phase, DNA replication occurs so that future cells will each have a complete set of the genetic instructions in the DNA. After DNA replication is complete, cells enter the G2 phase, where they continue to grow and prepare for cell division. At a checkpoint in this phase, the success of DNA replication is assessed. If all is well, the cell enters the mitosis, M phase. During the M phase, a complex series of events moves the DNA so that a complete set of genetic instructions will be sent to each daughter cell. The process of mitosis is assessed at a checkpoint during the M phase. Once this checkpoint is passed, the cell will complete mitosis as well as begin the cytokinesis C phase. Part or all of the C phase actually overlaps with the later parts of mitosis, so it is not a distinctly separate phase. During the C phase, the cytoplasm of the cell is divided, and two daughter cells are created from the original cell. When this process is finished, the daughter cells enter the G1 phase, and the cycle is complete.
Now, notice that it was mentioned in this animation in particular that cytokinesis, that last part of cell division, overlaps actually with mitosis a bit. It's not always distinct. And you have to remember, because we're talking about two different things. We're talking about mitosis. Specifically, we're talking about separating out the DNA. Separating out the DNA in the nucleus to two different nuclei, right, for the two different daughter cells. And we're talking about cytokinesis. We're talking about dividing up everything else in the cytoplasm, and eventually, obviously, pinching off the nuclear envelope and having two brand new daughter cells. Well, as we look at it in this cycle format, it looks like one follows the other, but in reality, cytokinesis really is at the tail end of mitosis at the same time within telophase. So there is overlap there, and that is an important distinction that we'll come back to. And then lastly, what I do want to talk about is how is this often regulated? Specifically, when we're talking about the human body, once again, this is more or less a human biology, intro to human biology class. We have to talk about what's going to regulate when a cell divides. Now, when we think about control and regulation in the human body, there are two primary systems, and we're going to go over this in anatomy and physiology. The main control systems in the body are the nervous system and the endocrine system. These two systems control the body, okay? And the nervous system does it through basically electrochemical signals, but the endocrine system specifically through chemicals called hormones. I'm sure you're somewhat familiar with this concept, hormones. Chemical messengers. So in this case, we have hormones. There are different types of hormones. In fact, they tend to fall under a couple different classes. We have some classes that are known as lipid-derived. Remember, lipids are hydrophobic, so they're lipid-derived hormones. The vast majority of those are steroid hormones. And then, whoops, I should make this line over here more. And then we have amino acid derived. Some of these are made from just a singular amino acid that has been modified, derived, whoops. Or some of these are small peptides and small proteins, small chains of amino acids. So the hormones fall under these two broad categories, if you will. And what we're going to look at right now is a steroid hormone. And these steroid hormones, we're going to travel in blood with carrier molecules, we're going to find out, because, well, they need to. They're hydrophobic. They all clump up in blood. So they're carried in these special carrier molecules. And then when they get to the proper cell, there are going to be they're going to enter through the membrane, because they are hydrophobic, and most of the membrane is hydrophobic. And there's going to be receptors on the inside, specifically they'll go all the way to the DNA. And now, steroid hormones, one of the things that they can do is they can tell the DNA to send a signal for the DNA to enter mitosis and replicate and grow. So then once again, most of our growth factors, are going to be these hormones to tell, once again, a particular group of cells within a tissue to grow. So we talk about what are going to be the control systems, the external control. We just had a little animation about internal control of this whole system of checkpoints and so forth. What about external control? Your body telling a particular tissue to grow. Well, this is how we have it. We have endocrine control of mitosis, cell division.
Steroid hormones are not water-soluble. They travel in the blood attached to protein carriers. When steroid hormones arrive at their target cells, they dissociate from their protein carriers and pass through the plasma membrane of the cell. Some steroid hormones bind to specific receptor proteins in the cytoplasm and then move as a hormone receptor complex into the nucleus. Other steroids travel directly into the nucleus before encountering their receptor proteins, not shown. The hormone receptor protein, activated by binding to the hormone, is now able to bind to specific regions of the DNA. These DNA regions are known as the hormone response elements. The binding of the hormone receptor complex has a direct effect on the level of transcription at that site. Messenger RNA, mRNA, is produced, which then codes for the synthesis of specific proteins.
So once again, this whole little section, once again, I'm a visual learner. Don't know if you guys are. I like the little animations. Animations went a little bit greater detail than the chapter did, actually. But the point is to get a big picture overview right now. We'll get into those details, but a big picture overview of interphase, of mitosis and cytokinesis, and how it's controlled. What are the basic steps, and kind of this general overview of the cell cycle. It's actually the next sections within the text where we actually get a bit more detail. So for right now, just go ahead and get this big picture of the cell cycle, interphase, and mitosis cytokinesis.